Today we are joined by Landon Porter. He is a PhD student in Hans Hoffman's lab at UT Austin. We actually overlapped there when I was a postdoc, which is how I know this fabulous scientist, and he's going to talk you through his research now.
Cool.
Take it away.
Thank you. Hi guys. So my name's Landon. The title of my talk is How to Knock Out a Balloon. V1a2 vasopressin receptor is a regulator of social dominance. This is one of my fish. I just put it on here. We'll see more of them later, but they're African cichlids. They're pretty colorful and they do some interesting behaviors. But at first I want to start out kind of broad and just define social behavior in kind of a really broad sense. So social behavior is quite varied across animals. And basically we say that animals are behaving socially when they're interacting with their conspecifics, so members of their same species. Social behavior takes many forms. This is an example of affiliative behavior. This is aggressive behavior, maybe two deer are fighting. Courtship displays, cooperative behavior, these ants are building a bridge, it's a pretty cool thing they do. And social dominance hierarchies, to name just a few. So social behavior is pretty varied. But regardless of the specific social behavior we're talking about, we see that it emerges from internal and external sensory integration. So what do I mean by that? Does anyone want to be brave and kind of tell me what they see going on in this photo? Anything else? Yeah? Bullying? Sure, aggressive behavior between these two, right? Anything else that's going on? Yeah?
The cheek fronts are raised up.
Oh, like specifically? Yeah, these baboons, they're aggressing and kind of trying to intimidate one another, right? Are there other baboons that aren't being so aggressive?
The two on the front, right? They're showing like cooperative behavior. So
Yeah.
this one's helping pick out.
Yeah, it's interesting. So in this photo, there's kind of these two things going on. You have this pair that is aggressing toward each other, maybe they're fighting in the back, but then right next to them you have this pair where they're doing grooming or affiliative behavior. And what's kind of cool is like this guy, or this individual I should say, has to be somewhat cognizant, right, of their social environment to understand how minute this is, that the person next to me is showing their teeth and being really aggressive, but that's not directed toward me. That must be directed toward the one behind me. So we say that this is the external social context that an animal exists in. This individual might be asking itself, you know, who is aggressing and is it directed at me? In this case, it's not. What about in this photo? You guys know what type of bird this is? It's a hummingbird? Yeah, I think it's a ruby red-throat hummingbird. Those are pretty common. Did we notice anything particular about this one? Does it look kind of, I don't know, stressed maybe a little bit? So the males have this plumage on their throats, hence the name. In the breeding season, it's really brightly colored. It's sexually dimorphic. But this particular individual, if you look closely at its feathers, it kind of looks like maybe it got into a fight recently. Maybe it's sick, kind of maybe not looking its best. So an animal also has to integrate its its internal state when exhibiting social behavior. This bird might be kind of asking itself, am I ready to mate, or am I too stressed out to try? Maybe I should sit this one out and try it next season. So when you have external social context plus internal state, you get a behavioral output. This is kind of a cute cartoon, not a cartoon, but an image I got offline of these dogs. Question for you guys, do you think that these dogs are fighting? No. How do you know? This one's biting this one's face. You think that maybe, you know, if I just describe this to you, like this dog is biting this other dog's face, you would immediately think, well, that's not very nice. But you can tell something subtle about this, right? You see this dog, his ears are kind of, maybe he's being playful. They probably know each other, right? This is an affiliative behavior. So the point that I'm getting at is that it's really complex, right? Is this appropriate? How do I respond? It depends on how I'm feeling, the nature of my social environment. So to behave socially, animals must compute decisions in real time, merging external cues with internal drives to decide what is appropriate to do and when. So this kind of begs the question, how is social behavior modulated within the brain? It's a pretty broad question. Are there even similarities across species that would allow us to formulate a general framework to answer this question? I mean, is it even appropriate to make generalizations? Maybe, I don't know if you guys think that birds have entirely different hardware than dogs and baboons? Maybe, to some degree, but what's really cool is if you kind of zoom in and look like at lower levels of biological organization, we see a lot of conservation. So hormones specifically, which coordinate context-appropriate behavioral responses across species, are widely conserved. Some examples, I'm sure you guys know what hormones are, and you could list off maybe even more of them than I could, but testosterone is kind of a common one, right? For example, testosterone promotes singing in male songbirds during the reproductive season. Oxytocin is another hormone that's been shown to promote bonding and parental care behavior. The list goes on and on. But, even though these behaviors are something that maybe we wouldn't do, chances are you guys don't sing in the breeding season and lick your children, right? So at surface level, you would think that these things are like wildly different than things that we do or other animals do. But kind of, I don't know, under the surface, right, testosterone, oxytocin, and the rest are kind of modulating something deeper, and that is these complex social behaviors. More specifically, at the genetic level, peptide hormones and their receptors are evolutionarily conserved. So again, just so we're all on the same page, I'm sure you guys know what hormones are, but basically they are signaling molecules that are circulated in the bloodstream. They're released by some cell, and they have target cells which express a particular receptor that bonds to that hormone, and that causes a response in the affected cell. When we look at these protein sequences of these receptors, we see that they are really conserved, right? So it doesn't matter if you're a bird, a baboon, a dog, a human, a giraffe, you have these specific hormone systems, and the receptors, sure, there's a little bit of difference, but the homology that exists in the amino acid and genetic sequences of these receptors is really quite substantial. This is kind of eye candy. It's what this actually looks like, right? So we have this, blob biology illustration, but this is a ribbon structure of a particular receptor. We'll come back to this later, but this is a vasopressin receptor, and this is the chemical structure of the ligand, the ligand hormone that activates it. So vasopressin is a particular peptide hormone that is really conserved across divergent taxa. And it's been shown to regulate social behavior in a wide array of animals. So how divergent, might you ask? So this is C. elegans over here, and studies have shown that vasopressin regulates reproductive behavior in these worms. It regulates foraging behavior in different species of ants. It regulates pair maintenance behavior in zebra finches, mate recognition in mice, and social dominance in wrasses. These are all very different behaviors, right, that are exhibited by these unique species. But at the heart of them all is that they involve cooperating and interacting with their conspecifics. And when we see behaviors that are like that, vasopressin is kind of the culprit, or at least in part modulating these sort of things. So the question that I'm asking for the chapter 1 of my dissertation work is how can we disentangle the role of vasopressin in social dominance behavior, or social behavior in general. This is a quote I included from Sidney Brenner. He's a Nobel Prize winner for physiology and medicine back in 2002. He said, choosing the right organisms for one's research is as important as finding the right problems to work on. And that's really true, right? If you have a specific question, you kind of need to think about what system would be best used to answer that question. And for me, in the lab that I work in, these fish are a really good model system for interrogating the types of things that we're asking and interested in. So these are the fish that I work with. At first glance, you might assume that this is a male and this is a female of this species. Perhaps they exhibit some sexual dimorphism, and the males are, colorful, as we see in a lot of species. But actually, these are both males. It's just that one of them is a dominant male and one of them is a subordinate male. So the dominant animals are characterized by being brightly colored, as we can kind of see. They're reproductively active and territorial, whereas the subordinate males are kind of bullied by these dominant males, and they're consequently reproductively suppressed, non-territorial, and they look kind of drab in coloration. But what's really interesting is that it's quite plastic. So the fish can, depending on their social context, right, and if there's a bigger fish in the tank or not, they will switch back and forth between these two, we say, behavioral phenotypes. So what's controlling that? This is an example of some behaviors that they exhibit. They're kind of complex. So let me walk you through what's happening here. These are the fish. This thing behind it is a terracotta pot. They love to hang around these broken pots. It's like their home. And basically what you're seeing here is these two males. They meet in the water column and kind of descend to the substrate where they exhibit this kind of ritualized behavior where they're basically delineating their territorial boundaries. So this guy's like, hey, you stay on that side, I'm gonna stay on this side, like we're cool. Although these fish are really good at individual recognition, sometimes it's kind of hard for us to keep track of who's who. So that's what these plastic like beads that you see on their sides are for. You'll see some more later. But yeah, that's some of the things they do. The vasopressin system has been shown to mediate behavioral plasticity in avertoni. So this is a micrograph from a paper that was published in 2008, where they looked at cell populations in the POA, or preoptic area. These are essentially just different cell populations, so different groups of neurons in that brain region. And what's cool is they found that the amount of vasopressin expression in these distinct cell populations varied with social status. I'm going to give you a second to digest that. So when we're looking at this cell population, so the smaller neurons in the POA, we call them the parvocellular neurons, we see that dominant males express more vasopressin than subs. But that's kind of flipped when we look at these large cells. We see that subordinate males are expressing more vasopressin than dominant males. So that's kind of interesting. Maybe it tells us that, hey, if you got vasopressin signaling in this cell population, you behave dominantly. And if you have vasopressin signaling in this cell population, you're likely to do so. But that's kind of only half of the story, right? I showed you this cartoon earlier. What I was just talking about was vasopressin, the ligand, right? So we were talking about the amount of vasopressin that is expressed in these cell populations. But it's really only half of the story, because in order to have any effect at all, the chemical needs a receptor. So my question that I'm going after is specifically involves looking at this receptor to see maybe it's similar. Maybe there's also some sort of like up or down regulation of the receptor expression in different brain regions across social phenotype. I know that this particular vasopressin receptor is extensively expressed in their brains, So this is a cartoon of one of these fish. They're great cartoons done by Caitlin in my lab. And this is a larger image of their brain. You can see some structures, medulla, optic tectum, hypothalamus. We basically take their brains and we slice them. We call it cryosectioning. I guess a little bit more on that later. But we slice them so that we can look at what's happening on the inside. So Huffman et al. Back in 2012 was basically set out to map the abundance of this V1a2 receptor, and they found that really no matter where you looked in the brain, this receptor was quite abundant. So they did this with IHC, immunohistochemistry, and in situ hybridization, just two different methods. We don't have to get into that. But they only did this for a couple of fish. They didn't really look at, is there a difference between males and females? Or is there a difference in dominant males and subordinate males? So that's kind of where my, question comes in, this broader literature, and that does V1a2 abundance and distribution reflect social status and behavior? We know it's there. What is it doing with respect to that? So yeah, I have some hypotheses. Basically that V1a2 expression within brain regions involved in social behavior will show differences. It's kind of vague, but I have some specific predictions regarding brain regions that I know to be involved in reproductive and aggressive behavior. So I have certain predictions regarding the amount of receptor expression that I would expect to see in those regions, but we'll see if that will work. Okay, so to answer this question, I have to set up an experiment. And basically what we do is we set up a fish tank. This is a fish tank. It's divided into two compartments by a clear plexiglass barrier. Basically just so I can get more fish per tank. That's really the only reason. But I call these the split tanks because they're split. On each side I have 4 males and 4 females. Out of the males, usually 2 of which become dominant and start defending these pteropods. The other ones are subordinate, and then the females just kind of hang out. After a couple of weeks, I let them settle into their new tanks. I take videos of them. So I take 10-minute videos of them in the morning to confirm social status. So basically, I need to identify who's a dominant male and who's a subordinate male. And I do this over the course of a day. You get the data back, and some of my undergraduates helped me a lot in scoring this, watching the videos, and counting the behavior events per 10-minute interval. So we see in these, these are, how should I say, categories of behavior. So for aggressive behavior category, dominant males show way more events per 10-minute interval than subs, as you would expect. Same thing for reproduction. The doms are courting females, they're doing all these displays and actually parasailing and spawning, whereas the subordinate males are not doing any of that. But then conversely, when we look at submissive behavior, that's like running away and shoaling with the rest of the group, we see that subordinate males exhibit that. So I have my 2 groups of fish. Now, once I've identified them, I take out their brains. And so we take their brains. Now I have dominant and subordinate brains, so I can go about quantifying V1a2 expression. How do I do that? So this is using IHC. How many of you guys, thumbs up if you are familiar with immunohistochemistry, or maybe I should give a thumbs. I see some wiggly thumbs or thumbs up. So basically, in a nutshell, I have this custom antibody, which is a protein that has a fluorophore attached to it, and the protein with the fluorophore is designed such that it binds to your protein of interest. In my case, it's the receptor. So all the places that you see fluorescence are basically where the antibody has binded to my protein that I'm interested in quantifying. So in the green channel, I have the immunofluorescence for V1a2 receptor. We'll see some better images of this, but you can kind of tell, as Huffman et al. Showed, it's pretty widely distributed. The blue that you're seeing is DAPI, which we co-label with. DAPI just stands for the nuclei, so it helps us differentiate individual cells because every cell has a nucleus when we're counting it. But yeah, and then I used the brain atlas, which kind of roughly approximates where these brain regions are. I did my best to find them on my slides, and I put them through this kind of analysis pipeline. So this is a little bit of a better image. This is the ATN. That stands for arterial tubular nucleus, which is the putative homologue of the mammalian ventromedial hypothalamus. Such a mouthful, but basically, if this was in your brain, it'd be in your hypothalamus. If we zoom in again at the ATN, we can kind of start to resolve. These are individual cells. They have a blue dot, which is the nucleus, surrounded by this green glowing. That's how we identify them. At first, my advisor was like, "Yeah, Landon, like, why don't you just have all of these different brain regions for all of these fish and count by hand all of the cells? You can do it. You know, you gotta work hard as a grad student." I was like, "Geez, I really don't want to do that." So fortunately, you know, advances in technology and machine learning and all this stuff, I found this software called CellPose 2.0, which basically uses deep learning machine algorithms that I don't quite understand to automatically segment your particular data. They have different models that you can use. I don't know if any of you guys do like cell counting stuff. I would totally recommend checking this out. They do different models, but you can actually train your own model. I didn't do that because it worked just fine, but if you have like, you know, weird data or some weird cells, you can train it to count your data. But I just fed it these processed images of my cells, and it counted them. It gives you an output, so it's pretty fast. And so that enabled me to do this for a number of different individuals and for a number of different brain regions per individual. And what do we see? When I was doing this, I thought to myself, I'm like, there's no way. Like, there's no way that I'm going to be able to recover a biological signal, especially when I was doing this stuff, but trying to, like, okay, where do I draw the line? Like, how am I consistent across sections? I thought that I'm just like, well, I'm just gonna like have no result, and then I'll just be done with this chapter and can move on with my life. To my surprise, like it must have been like a pretty strong signal to still show up, despite, you know, the differences in how I like felt that the ATN looked one particular day. We still recovered that if you look across dominant and subordinate individuals, we see that the amount of V1a2 expressing cells is much higher in DOMs than subs in the ATN. Maybe, although not statistically significant, but a similar trend in the TPP. The POA, which is that brain region we looked at earlier, looks like that's significant. IL. I'm in the process of doing this for 2 more brain regions, and I hope, I mean, you know, it is what it is, but my hope is that maybe I find a brain region that actually shows more in subs than in DOMs. That would be cool, but we'll see. But this is a nice result. This is like chapter 1 of my dissertation, and I'm looking forward to publishing of it. Like, yeah, we see that not only does the ligand vary its expression across DOMs and subs, but also the receptor is doing something cool in a brain-specific manner, brain region-specific manner. I also showed, and this is pretty cool, so when we graph the amount of aggressive behavior on one axis with the cell count in a particular brain region, I see a trend here with DOMs, such that the more ATN you have, the more aggressive you are. The more V1a2 you have in your ATN, the more aggressive you are, the more reproductive events you exhibit. Same with the TPP. It seems like the more receptor you have, the more aggressive and the more reproductively active you are. So let me ask you guys a question. Based on what I've shown you so far, what do you think a fish would behave like if they didn't have any receptor expression at all? If I'm saying that like more receptor means more behavior, no receptor at all, I don't know, it's kind of reasonable to maybe assume or hypothesize that they're not going to be aggressive or reproductive at all if I take away the receptor. Stay tuned. So my next question is, what is the function of V1a2 in regulating social dominance behavior? How do we go about, okay, so I can, do IHC and look at the differences in expression and, correlate it with behavior to get us some sense of what's going on. But really, in order to answer the question of like, what specifically is this receptor doing in these brain regions, we need to be a little bit more precise. So we can probe the function of V1a2 using CRISPR gene knockout. You guys have probably encountered this before. I'm just going to go like a rough overview. How do we knock out a specific gene? Well, we basically design custom guide RNAs, which are short DNA sequences that are complementary to a particular part of the genome that we're interested in targeting with CRISPR. So how that works, you have your guide RNA that you co-inject with the Cas9 enzyme. The way I explain this to my students is basically the Cas9 is a blind pair of scissors. It can cut DNA, but it doesn't know where to cut, and that's why you have to give it some instruction in the form of guide RNAs. It's like, hey, blind scissors, come here and make a double-stranded break. So that's what we do. We co-inject the guide RNA with the Cas9 enzyme into single-celled embryos. How do you get single-celled embryos? This is a video I took in Maryland, University of Maryland, when I was doing the injections. I might play this a couple times, but you can see the female lays eggs, and she looks like she's eating them. Wait, see? She picks them up and puts them in her mouth. Whoops. That's because this species is actually, they're mouth breeders. So the females keep the babies in their mouth for 2 weeks while they develop, and then they spit them out. I think there's one on Nemo. Have you seen that? Or it's like the kids, yeah, that's this. And so you might, if you're thinking about this, wonder, okay, well, if the female lays the eggs and puts them in her mouth directly, how are they fertilized? This is the only species that I'm aware of that reproduces via oral sex. And it's kind of underhanded, the way that the males do it. So if, I don't know how to pause this as it's going. You can kind of see the males have these spots on their fin. You can see my cursor. He has these spots on his fin, right there. You saw them? They're yellow, and they're essentially the same size and shape as the eggs that the female's laying. So he basically puts his fin on the ground and is like tricking the female into like, wait a minute, I thought I just picked those up. So she tries to pick up eggs again, and that's when he fertilizes them in her mouth. So very interesting reproductive strategy for these guys. But yeah, I got to see that in the morning. So basically, after the eggs are fertilized, we flush them out of the female's mouth. It's better because then she can start eating and stuff again. But it's helpful because I want to get the single cell stage, right, for my injection. So we immediately take them out and put them in a gel. So this is a really squishy gel that I used a paintbrush to kind of position them in. And then I injected them with this serum that contains the Cas9 and the guide RNA so that the CRISPR will target the V1a2 gene that I'm interested in, right? Just to be a little bit more precise, so genes have different exons. I targeted at the very beginning of this gene. So this is the start codon. This is the first exon of the V1a2 gene, and my 2 guide RNA targets are like, I don't know, 100 base pairs deep into the gene, so very early on. And the idea of having 2 guide RNA targets is you're going to have a break in the DNA here from one of them, and you're also hopefully going to have a break in the DNA here, and when the endogenous repair mechanisms try and fix that broken chromosome, that chunk of approximately 50 base pairs in between these 2 cuts might get removed. So I would get a large 50 base pair deletion in my gene, which results in a frame shift that takes out its function. But yeah, so after I do that, I kind of watch them develop. This is an embryo, right, developing. I think this is day 4, maybe day 5. You can see its eye starting to form. These black dots are actually melanocytes that are going to migrate into the animal as it's growing. And you can kind of see if your eyes are good. If I can play this again. This is its heartbeat. It's kind of cute. I'm like watching them develop and grow. I'm like, oh my god, it's so cool. I feel like a father. Okay. Back to my question that I asked you guys. What do you think would happen? So I did all this work. I CRISPR'd their gene, right? And there's some other stuff that I'm not even including, but I sequenced them. I know that it's knocked out. It's all good to go. My hypothesis was that if I took away this gene that is apparently correlated with reproductive and aggressive behavior, that they shouldn't be dominant, right? Well, then they grow up. This one, super colorful. He's got his eye bar on. He's like being a little bully, like chasing all these other fish. I'm like, dang, did I mess up? Did I do the wrong gene? So I tagged them all with these tags. I sequenced them 3 times. And I'm like, yes, it's actually gone. It is a frame shift mutation. So why are they still behaving with it? Did I just waste 2 years of my life? So I'm talking to my advisor, right? It's where do we go from here? And it's kind of okay, a lot of times you have a hypothesis or a prediction, but biology being biology, right? It's oftentimes a little bit more complicated than we even kind of foresee. So he basically is okay, well, let's put them in this experimental paradigm where we look to see if maybe there's something more subtle going on. Maybe it's not an on or off switch, but something in between. So let's just see what happens. Here's a different experimental paradigm, kind of same thing as before. I have a fish tank, I split it up into different compartments. There's males and females in each. In the center compartment, I have a focal male. So he's gray right now because he's subordinate. And the idea is that you put him in an environment that has a larger fish, he's going to stay subordinate. But if you remove the dominant animal, at least in the normal non-mutated fish, we do this a lot, and this guy's oh, now's my chance. I can flip on my colors, and I'm going to start reproducing and defending this territory. The technical name we use for this is a bullyectomy. Remove this guy. And then, we quantify the behavior change of this particular focal male as we change his social environment. We do that taking videos, like I did before. So what do we see? What does that look like? You guys see this okay? It's kind of dark. Should I turn off the... Yeah, I should have done that video. I can see it. So this is the... Thank you.
That's too much.
You guys are going to go to sleep? That's all. No, I'll turn it back on in a second. But, so this is a wild type fish. So this is one that isn't mutated, and this is just what they kind of naturally do. So this is pre-bullyectomy. What do we see here? We see this guy. Look at how big of a shit he is. He's aggressing at his neighbor. These guys, the other fish, what are they doing? They're literally terrified. They're don't look at me, please, right? This is the other male. This is our focal male. He has his nose in the corner, right? Biding his time, waiting for, I don't know, a predator to come pick this guy off so that he can do his thing, right? And we see that, the predator being me fumbling around at 4 a.m. before the lights come on to fish that guy out. The next day, that fish that was in the corner over here not doing anything has turned on his colors, has an eye bar, is posturing, being aggressive toward his neighbors. This is a very plastic thing, and this is why this is such a good system to work on, is because we can kind of see these changes happening in real time. It's fun to watch. So now what happens when we do this same experimental setup, but we do it with a mutant? So this is the mutant male. I didn't show you the day before, but this is he had a bigger fish in here suppressing him. Now he doesn't, right? So if he was normal, you would expect this guy here to have more coloration, an eye bar. He should be like, you know, trying to get at this guy, maybe courting the female over here, but he's not. He's just hanging out. And it's not that he's sick or something. I mean, if you keep watching it, he starts to like, you know, peck at the food, he swims around a little bit. He's just utterly like uninterested in doing these like dominant reproductive displays and stuff. So that's interesting. Like, wait, what the heck? I know that some of them are dominant, but when I put them in this very like specific experimental paradigm and they start out subordinate, it's almost like they can't get their act together like a wild type fish does. So I'm going to take you through, I've done this a number of times, and this is a little bit preliminary, but this is what I see so far. So these are just different categories. The big difference is that I have the receptor knockout population, and then you can think of these as the same. They're just from two different like backgrounds, like genetic backgrounds of fish, but they do the same thing. They're wild type. And what do we see? Day -1, we're talking about the total aggressive behavior exhibited by that focal male, the one that was sticking his nose in the corner. He wasn't doing anything, right? The normal fish, they don't do anything. But on day 0, when we remove the dominant male, we see that the amount of aggressive behavior goes up. It spikes. Makes sense. Dominant male is removed, we did the bullyectomy, so that subordinate male is now being like aggressive. And it kind of like tapers off a little bit. They're really aggressive on day 1, and they kind of establish their territory, and then they switch from being aggressive to more reproductively active. But that's what happens with the wild type fish. Now let's look at this population of mutants. Well, I had a number that started out subordinate, and then on day 0, they kind of didn't do anything. That's like the video that you saw before. Just wasn't interested. By day 1, maybe a little bit. Some of them are kind of starting to aggress. Day 2, I don't know. They're just kind of lazy. And that's interesting because I know back when I showed you that video that these guys are capable. They're certainly capable of being dominant, but when I put them in this experiment, they're kind of not able to go from subordinate to dominant in a way that we think is normal. But I'll show you like one data point that I think is probably the most interesting out of all of this. And that's this guy. This particular individual was exhibiting aggressive behavior before I took out the bully. And if I had left the bully in there, he probably would have died because he would have lost the fight because I paired them with much larger fish. But he's like, I don't care, I'm gonna fight him. This guy was a dominant male before I put him in the experiment, whereas these guys were subordinate before I put them in. So what we think this means is that rather than being an on and off switch of like, if you have the receptor, you know, expression in your brain, you can be dominant. It's more like a control mechanism for the plasticity. I didn't take away their ability to become dominant. I took away their ability to change back and forth between phenotypes. So it's kind of more subtle, but maybe even more interesting. So yeah, that's kind of the conclusion that I'm led to thus far by my data. It's like if you develop, if you're a fish, you're a juvenile, and just, you know, a roll of the dice, you develop as a dom or a sub, you're stuck that way. And no matter if I put a bigger fish in your tank or take him out, you're not going to be able to integrate the change in your social environment to behave appropriately. I conclude that V1a2 signaling is needed for social transition, not social dominance itself. So that's kind of cool. Future transcriptomics experiments will show what's going on in the brain as a result of V1a2 knockout. I'm excited for those in my chapter 3. Yeah, it'll certainly still be interesting. So that's kind of it. This is my lab. Thanks to the Hoffman Lab and everyone there that helps me. I have some collaborators in Maryland that I did the injections with, and then Rick Klein at the University of Texas at Rio Grande supplied the antibody. So yeah, I'm doing this about those. Thank you. Put the lights back on. And then I'll take any questions. Yeah.
I'd like to know
That's a, like, I feel like I always say this, but that's a really good question. Because, right, it's kind of, we call them alternative reproductive tactics. Alternative reproductive tactics. So basically, if you're a small male in a pond with a lot of larger males, you don't really stand a chance of reproducing until you just hang out, you grow, you do you, work on yourself, and then maybe a couple years from now, I'll be large enough to actually, compete and defend a territory. But then you're like, okay, well that's one strategy. Why would there even be turnover at all? Because you're thinking that, okay, if there's a bigger fish, chances are, like, I'm never gonna have a chance. What do you think in, like, the natural environment of these fish would enable, like, a smaller fish to, bide his time and then eventually take over a territory?
I'm thinking more of a predator.
Yeah, right. Being a dominant male comes at a huge cost. You can be dominant and you can reproduce, but at the cost of, like, you've got these conspicuous colorations, and so a bird's gonna come and swoop you up in, a week or a month or so if you're lucky. So it's kind of like you have to bide your time and only become dominant when you're sure that that's gonna result in some sort of adaptive advantage in your situation. Yeah, good question. Any other questions? Yeah.
Say you were to address the endocrine aspect of it, and you put so much testosterone in all the fish.
Mm-hmm.
Would that change the dominance or the dynamic, or would the dominance become more dominant?
If you injected them with testosterone, you're saying. So, yes, to, like, your thoughts are correct. If you inject a dominant male with testosterone, it becomes more aggressive. It increases their behavior, but not a subordinate male. So you can do hormone assays. Like if I in this situation, I'll take pre. This guy. You remember this guy who had his nose in the corner, right? He's a pretty big fish, right? He was dominant. Like when I took him out of his home tank and put him in here, like he had to like kind of switch up. If I did an ELISA or a hormone assay, he would have high testosterone. Kind of that taper takes like 2 weeks to like actually like set and like slowly say like fully transition into their respective phenotype. But at this point in the experiment, yeah, if I tested his testosterone, he would have just as much as this guy. But for some reason, there's a switch in their brain that like overrides that, and they don't. Makes it very weird. Yeah.
Is there anything with size, like could a smaller fish be more aggressive if that's like bigger fish or something?
So there's a little bit of literature on the different like color morphs behave slightly differently. It's like the yellow ones are actually more aggressive than the blue ones, which is kind of weird. But generally, like 99 times out of 100, the larger fish is going to become the dominant one and suppress the aggression and reproductive of the smaller one. Yeah.
Sorry, I'm not sure if I missed it or not, but I was wondering, like, what would be the behavior of these subordinates? Like, if they were placed in with dominant and the female, and one with just the males, the dominant and the subordinate. I mean, like, one we have females and one we don't.
Sure.
So would they like try to make impression and try to be dominant or not?
Yeah, that's funny. These fish are actually really smart, right? You kind of think of goldfish as having like a 3-second memory or whatever, but these guys, they're sharp, dude. They sometimes you can see they have this like sneaker, or we call them intermediate males, and they like pretend that they're a subordinate male as long as this guy's looking, but when he gets busy, like he's in a combat with his neighbor over here, sometimes he'll turn on his eyebar and start like courting females, and then this guy like comes back and he's like, nope, I wasn't doing anything. It's cool to watch, and that they are so hyper aware of like their environment that it does affect their behavior. But to answer your question specifically, in the case of if I just put a population of males in here, they would still like aggregate together, but you wouldn't see much reproduction or aggressive behavior amongst the shoal of subs.
dominant fish slowly loses its elasticity to transition back into the southern dom if it were to stay with the bully for extended periods of time?
In like a normal setting where I didn't knock out their genie, you're asking if, you know, this guy's been a sub for 3 years or like, you know, a considerable fraction of their lifespan, do they still maintain this capacity to transition?
Or even in a laboratory setting
Yeah.
where it just spends a longer amount of time with the bully?
I think, I haven't tested that specifically, but I think the idea is that they can do it no matter, how long. As soon as the opportunity comes about, they're pretty willing and able to do it no matter how long. Yeah, good questions, guys. Oh, yeah, sure.
Do you think the lack of plasticity, so like the lack of transitioning on the social security, or is it due more to that lack of monitoring of the social context? Like, do you see deficits in other social monitoring?
Sure. I think that's an empirical question that we're certainly interested in. I mentioned for my chapter 3, I'm doing the transcriptomic pipeline where I'm basically going to sequence the like the RNA to survey gene expression in these different brain regions, and depending on the differences, maybe if we only see differences in a brain region that we know is involved in like social sensory integration, then maybe that's the hang up. Or maybe that brain region is relatively unaffected in terms of a gene expression profile, and that a different region that's involved in aggressive behavior might be like the culprit, I guess, for this phenotype. Yeah. That's cool. Yeah.
So we had this mutant outlier, right?
Yeah.
Do you know at what point he became dominant?
In terms of his development, I do not. I mean, I watch them, and usually they go through this juvenile phase. We call them like mini doms. They're super cute. They're like little teeny tiny, but they're the biggest fish in their tank. So even though they're really small, they like turn on their eye bar and they start, you know, being like a little aggressive. It's like Chihuahua or something. It's cute, you know. And I guess that usually takes like 3 months before we start seeing that behavior. So probably my guess is that one particular individual, after 3 months or so, just developed into a dominant male as the fish was growing and kind of got stuck.
So was it the largest among the population?
Yeah. Maybe I just ate more and grew a little bit. Yeah.
I have another specific
Sure.
question, but
Yeah.
if you were to group a bunch of small dominant fish in a tank against the bully fish, do
Yes.
these fish species exhibit any grouping differences in behavior?
I don't think so. Not that I've seen. Some species definitely, you're thinking that like, oh, we'll team up and take this guy out. That'd be cool, but I don't think they do that. Yeah.
So I have a question about vasopressin in other animals.
Sure.
So taking this beyond fish and other social animals that
Yep.
might go through male dominance
Higher.
behavioral
Sure.
changes. I'm thinking lions, but that's
Yeah,
where
or
I'm
mice. There's a
going.
lot of, yeah.
Sure.
Uh-huh.
Do you see similar, or is there any literature on other animal systems that
Yes.
parallels this?
Yes, there is quite a bit. Let me think. So, I hate to just like pull this card and say it's complicated, but it is kind of complicated in that we see that vasopressin is kind of doing different things in different species, and even in different directions. You might see that in my fish. In my thesis, I'm saying that okay, the more receptor expression you have, the more aggressive you are. In a mice system, we might see the opposite, that actually it's like the less you have, the more aggressive you are. So kind of making these generalizations as to what vasopressin is doing at that sort of scale is difficult, but that being said, underlying it, it's like, well, it's still involved in their dominance hierarchy and maintenance of their social behavior just in kind of species-specific ways. So yeah, kind of taking an integrative approach, I think, is really informative. And you have to look at that. You can look at this through the lens of a comparative biology approach, but yeah, I don't think I have a specific thing that it does across species. Anything else? Oh, sorry.
What happens with the knockout females?
I do have knockout females. I haven't tested them. This is an anecdote, but it's kind of annoying because I want to establish a large population of these fish. And what I'm struggling with is that I didn't talk about the genetics and the Punnett squares and the crossing that's involved in this, but basically I only can get reproduction with the heterozygotes. The homozygous females don't seem to brood, or like, they exist, they're healthy, they're there, I feed them a bunch, they grow, but I have yet to see a female mouth brood. So something might be going on there, although I haven't tested it.
I have I think might be a follow-up question, which is, it sounds like there might be an inability for these fish of both sexes to really not know what's going on in their social context.
Yeah.
I'm interested to know, like, does that potentially only go one way? Because you had subordinate fish that you put a dominant fish in and took out. What if you have a subordinate fish and then you put in a smaller fish? What do they do?
That's kind of like what this was, inadvertently, right? Like, I set up this experiment with the idea of like, okay, I'm gonna have a bunch of subs. I give them a week to kind of sub out, like they limp out. If they get beat up by the bigger fish, then they become subordinate. That guy didn't, right? He probably was not very healthy, but yet still trying to be aggressive because he couldn't go the other way and become subordinate. So I think it is very much an inability to transition in either direction, more so than it is to exist as a dom or so. Yes.
So to like follow up on that follow-up,
Yes.
Is it possible that there is some sort of like critical window for these fish? Because you say like as they're developing, they are sensitive to their social context,
Totally.
and then they just stop being at some point, right? Or am I misreading?
I think maybe if there's, how would this work? If you're the first one, if there's no one in your environment, you hit puberty early, whatever the case is, maybe you ate a little bit more, just by chance, right? You become the largest fish in the tank and there's no one to suppress you. Maybe you can become dominant then. But as soon as you're put in an environment thereafter where you need to change or do something, I think that they're kind of incapable. But it is, an open question, like how exactly this is working through you. Yeah.
So follow-up question on that is, when you do your
Transcriptomics, yes. Yeah.
Are you going to be looking at the dominant, the submissive, and the females as three separate potential, or is that too much for a breed?
I am interested in looking at the females in this sort of context because they also, although not as like colorfully, like they do exhibit dominance hierarchies like amongst themselves, and they have eyebars and kinds of things that's just more subtle. So I'm interested in looking at that. I think that would be a nice undergraduate project, and I have someone who's interested in that. But as far as the transcriptomics go, it's really expensive to the sample. I mean, the sequencing is like $13,000 or whatever for 6 or 7 of them. So it's kind of a lot, and I have to be careful with which ones I think would be most interesting or informative. But what I'm basically doing is a time course set. So I'm taking DOMs and subs at different time points, at day -1, at day 0, over the course of the experiment, to see how, in a normal fish, their gene expression profile changes with time. But maybe in the mutant fish, it changes in a different way in the temporal dimension. And that might be what's causing this difference. So.
If you have an extra $10,000, I recommend doing the
females.
of course. Mostly to help you in breeding, right? Because if they have different profiles that prevent them from breeding, that may be informative for why you're not able to, rather than spending the next 5 years.
Yeah, and then right. I mean, the heterozygotes, they breed, and then it's like a Mendelian, where 25% of the offspring are homozygous for the mutation. So I have to just screen them and pick out the ones I want, which is kind of annoying, but I have a nice system in place for that. But yeah, ideally, oh my gosh, it'd be so, I could just like leave them alone, let them do their thing, but yeah.
So you have a GoFundMe that you
Maybe, yeah. Well, these fish will exist well after, I mean, they're gonna stay in, you know, Hans's lab, so if other people want to do research on the females, like after, I think that's certainly, and in fact, probably going to happen, I think, with, yeah, in the future. Thanks, guys.